primary voice of reason in understanding and interpreting neuroimaging studies and their limitations, and is a great voice and advocate for responsible science in the neuroimaging community. So and he has so many honors and awards, I can't list them all, but the one that I think of most fondly is that he was one of the early Wiley Young investigators from the Organization for Human Brain Mapping, which is given to the Outstanding Young Investigator of the Year. I can't remember how long ago that was, like 10 years ago or 10 something.
10 years ago.
Gosh. The winners of this award have gone on to do other great things. One of the earliest Wiley Young investigators won the Glass Brain Award at OHBM, and I'm sure that we will see the same for Russ in his future. So it's with great pleasure that I introduce Dr. Russell Poldrack.
Thank you. Thank you very much. It's always fun to be back here at UCLA, and I'm really happy. I mean, I'm sad that the course is ending this year after 10 years, but Mark has done an incredible job of sort of building it after I abandoned him a couple years in, and I think it's a testament to him as to how successful it's been. So what I want to do today is talk about how we might think about what fMRI can tell us about the structure of the mind. I'm trained originally as a psychologist. I'm ultimately interested in understanding, how is the mind put together, right? And ultimately also, how does this piece of meat, do the computations that let us do the crazy things we do with our minds? So let's just start out with a sort of a thought experiment. So this is a plot of the cumulative number of publications mentioning fMRI in PubMed over the last 20 years. And you can see it's now up above 40,000. If you get these weekly reminders of new papers, it's on the order of about 100 papers a week now. No way anyone could keep up with that. And so, if you look back, when I came here, Susan mentioned in 2002, it was down around 4,000, right? So we have an order of magnitude more papers now than we did back then. Do we really know an order of magnitude more about how the mind is implemented in the brain than we did 14 years ago? And maybe more importantly, if it were to get up to 90,000, which at this point is going to happen within a few years, will that tell us twice what we already know if we keep doing the same kind of thing that we've been doing before? So what are the how do we answer that, right? Well, you might imagine there's some questions that we'd like to be able to answer. You'd hope that all that money, all that time and effort, all the formerly trees, would actually allow us to answer some important questions, like what does insert your favorite brain area do, right? What's the function of a particular brain area in terms of cognition? Or how is some particular psychological function implemented in the brain? So let's just take an example. What does the anterior cingulate do? I'm not going to ask for questions from the audience because we could spend an hour on that. But if you search for anterior cingulate and fMRI in PubMed, you find about 3,600 abstracts. And so just under some really conservative assumptions, that's over $20 million just in data collection, not to mention all the time of people that went into to doing that. So how might we answer the question of what the anterior cingulate does? Well, one way you might do that is to say, what are the functions that are associated with activation of the anterior cingulate across the literature? How many of you are familiar with Neurosynth? Okay, so Neurosynth is a database. You can get it at neurosynth.org. It's completely open. That basically has activation coordinates automatically pulled from over 10,000 papers. And basically what you can do is say, I want to know what regions are generally active in papers that mention some particular term. And so these are meta-analyses done using that toolbox for a number of different terms: maintenance, pain, phonology, interference, difficulty, conflict, errors, attention. You can see that they all activate the anterior cingulate. And in fact, if you look across all the studies, all those 10,000 studies in Neurosynth, on the order of about 20% of them have anterior cingulate activation. So it's really hard to imagine that we're going to be able to say anything about what the anterior cingulate actually does using this sort of strategy. And there's a really old paper that I think nicely lays out the problem with this strategy. It's a paper by Alan Newell about a very different sort of problem. This was at the very beginning of cognitive psychology, And he was sort of talking about the fact that, there were a lot of different psychologists asking, is attention, early or late selection? Is processing serial or parallel? He titled his paper, "You Can't Play 20 Questions with Nature and Win." And I think you can think about, and I actually stole my kind of first thought experiment about, where will 40,000 more papers get us? I stole that from him. The point being that, doing more tasks and asking, what lights up when people do task X and task Y and task Y prime and Y prime prime and so on, is not necessarily a good strategy for actually figuring out how the brain works and what particular parts of the brain do. So recently, Tal Yarkoni and I, in an annual review chapter, argued that instead what we need to do is figure out how to actually establish what we call selective inferences. The idea being we need to figure out what can you actually predict from the engagement of a particular region or set of regions or pattern of activity, rather than just saying, what's associated. And the point being that when you can establish that more selective relationship, it gives you a better handle on what are the actual processes that are predicted from the engagement of that system, rather than just what's correlated. So we started doing this a few years ago. Our first paper, published back when I was still here, basically took a bunch of data we had collected in my lab on 8 different tasks across, I think, 130 different people. And we basically just said, if we take each of those tasks, and they varied from a language task and a decision-making task and a motor task, a really, really widely varied task, we basically said, for each of those tasks, let's get an activation map for the task versus some sort of resting baseline, and just ask, can we classify what task a person was doing based on their brain activity when we've never seen their brain before? So the novel thing here was actually predicting to new people. Most of the work that had been done up to this point had been basically training people and then look at what you can predict about that person using different data from the same person. And it turned out we can do pretty well. We can get on average about 80% accuracy at predicting on this 1 out of 8 classification problem. That's the chance there is around 12%. We've since pushed it to even more tasks, right? So we can, in the more recent work using the Open fMRI project, we've looked at the ability to classify which of 26 tasks a person is doing. And we show that we can get reasonable accuracy around 50%, where chance is now down well below 10%. Importantly, it seems like we're actually using pretty low dimensional information about the brain. What you see here is the number of ICA components that we projected the data down into, and our classification accuracy. And basically, once you are certainly once you're at 100 components, so you've taken those 220,000 numbers from a person's data set and collapse them down into 100 numbers, and you can basically do just as well as you can with the whole brain data. And you can do almost as well even with 10 components, suggesting that what we're really looking at is kind of combinations across tasks of fairly gross, low-dimensional sorts of patterns of activity. But ultimately, I don't care so much about being able to tell you whether you're doing a lexical decision task or a stop signal task or a insert your task here. What I really care about is decoding what are the functions that are ongoing in your brain, sort of apart from any particular task. And the problem is we need to know, before we can kind of predict what the functions are, we need to know what's the possible set of functions we can predict from. And so I'm going to take you on a little aside. Pretend you are chemists. And you would hope that there's a certain set of questions you would hope chemists can answer. One of them is, what are all the enzymes? Chemistry does a lot of stuff with enzymes. You'd hope they can, if you ask them, tell me all the enzymes, that they could do that. And it turns out that they can. This is Paul Boyer, who's actually on the faculty here, who started a number of years ago writing a set of books called The Enzymes. It's now up to like 40 volumes, and it tells you what are all the enzymes. Furthermore, if you go to the Gene Ontology, you can find out not just what are the enzymes, but where do they sit in particular biological pathways? What do they do, right? Now, you might hope that we would have something like that for psychology. Like if you pick up a cognitive psychology textbook, would it tell you what are all the things that the mind does? And the answer is maybe at a very gross level, but not at the level that you need to be able to actually do the kind of analysis that we want to do. And so it leads to this question, there's a whole organization, we just came from the meeting, called Organization for Human Brain Mapping. The idea is we're mapping something onto the brain. What are we mapping onto the brain? interestingly, back in the day, you often saw things like this in papers where people would say, here are my 4 different tasks that and the subtractions that I'm doing, and these are all the psychological processes that I think are being tapped by all those tasks. And then you could, in this case, this is from Kathy Price's famous conjunction paper, you could say, we're going to find a conjunction across those different contrasts that only taps into one of those cognitive processes. The point being that, has anybody seen this in a paper since 2000? I haven't. And the point being that I think we've kind of fallen back on the need to be really sort of deep in terms of how we analyze cognitive tasks. So we started a project when I was here. This is particularly working with Bob Bilder, called the Cognitive Atlas, that sort of aimed to do this. You can find it at cognitiveatlas.org. If you're interested, sign up. We'll give you an account, and you can actually get involved and contribute knowledge if you want to. The point of what Cognitive Atlas does is sort of lays out what are all the things that we think the mind does, and how are they measured. So we have 2 separate sets of constructs that are in our database. One is what we call mental concepts. These are sort of things that psychologists dream up. They're latent in the sense that we can't see them. We can only see what their putative effects are. Those are things like cognitive control or working memory updating or set shifting and so on. And then we have tasks that we think measure them, right? Those are the things that we actually do in order to try to manipulate things that are in people's heads. And so what we do is we lay out we have really sort of separate databases. One that lays out, what are all the things we think the mind does. The other lays out what are all the things that people do to measure it. And then we basically define what are the relationships. That's the really important part of this. We say, for example, this particular measurement on the stop signal task measures some particular concept called response suppression. So one thing that we are very careful to shy away from is kind of mixing those two. So if you ever hear somebody use the term working memory task, right? That's really a theory about what that task is measuring, right? The task is a delayed recognition task or a some other sort of task that's measuring something people do. Whether it's actually measuring working memory is a hypothesis that has to be tested and confirmed or denied. So we try to be very clear about the distinction between those in the atlas. So we've spent a lot of time over the last 10 years trying to build this database to actually have knowledge that can tell us what do we really think it is that we're mapping onto the brain. It still is far from where we'd like it to be. It's a big problem. And my lab only has sort of expertise in parts of the whole domain. But here's sort of the ultimate goal, right? The ultimate goal is to be able to do completely automated ontology-based meta-analysis. The idea would be you have a bunch of data, a bunch of brain imaging data. It could also be behavioral data or genetics data or whatever other kind of data you have that are related to tasks. You have those data, you have your tasks, and then you have this ontology. That's what this tree is that basically says, for example, this particular contrast on that task measures this story comprehension, this particular contrast on the lexical decision task measures lexical processing. And then we can use our knowledge about the relationships between those to actually infer something about what particular brain systems are supporting which particular cognitive functions. And as you'll see later, we can also use it to think about whether certain cognitive functions might be sort of better or worse motivated or justified by the data. So this is the goal. In a sense, this is the goal we've had for 10 years. The problem is, where do the data come from, right? We need lots of data to do this. We still don't have anywhere near enough data to do it because we need lots of data from lots of people on many, many different tasks that span the entirety of cognition, basically. We certainly don't have that. What I'm going to show you is kind of a small snippet of the domain of cognition. We've been engaged in data sharing for a long time, and I didn't get started in data sharing because I'm some sort of lofty pie in the sky thinker. I got started in data sharing because I wanted more data, and I knew my lab couldn't create enough data. The hope was that I give my data to the world, other people also give their data to us, and ultimately we can put it all together to do bigger things. It's took a long time, but it's finally starting to happen, even though, again, we still have nowhere near the amount of data that we need. The big advancement has really been in developing ways to make it easier for people to share data. So we started with the Open fMRI project, which was about sharing complete raw data sets. That's a real pain, right? Because they're big, there's lots of metadata you need, details about the task, and so on. And it turns out that for a lot of things, you don't actually need that, right? All you need is the map, the statistical map. If all you care about is what's in the group analysis, all you need is that statistical map. And so a couple years ago, Chris Gorgolewski and my group developed a new project called NeuroVault, which basically makes it really easy to share statistical maps. The idea is you go on and within a couple minutes, you can basically upload your map, upload at least a little bit of metadata about sort of what it's about, and then you can view it online, you can share it with people, it can be private or public. It's actually a really useful tool just for kind of sharing data with colleagues when you have a paper and so letting them look at it in more detail. You know, when we write papers, we make these figures. There's like one slice with some activation on it, which is such a, a kind of a denuded representation of what's going on across the whole brain. This lets you give it to people in such a way that they can actually look at the data in a much richer way. So what we've done is basically gone through, taken some data sets from NeuroVault and gone through and annotated them using the Cognitive Atlas. And what that means is basically we sit down with a data set, with the paper, and we basically try to figure out what were they studying? What were they trying to measure with, for example, this particular contrast comparison of parametric pumps versus control parametric in the balloon analog wrist task? What psychological functions do we think are being tapped by that particular comparison of conditions? We put those into the Cognitive Atlas. We think it measures risk and reward anticipation and response execution and other things. We do this for each task. This takes a lot of time. Sometimes we'd spend an hour on a particular task and not even come to a final conclusion about it. So we've done this for 93 data sets. That's way too little data to make any big conclusions about. So really what I'm going to show you is a proof of concept more than anything else. It's a proof of concept for why these kind of data and this kind of annotation using an ontology might be really useful in the future. So the first question we can ask is just, does the ontology relate at all to the brain data? So we have a bunch of tasks, and for each of those tasks we have an activation map, and we can compare those activation maps. We just compute the Pearson correlation between them. Then we have our mapping of the ontology, right? We said that for this task it involves visual sentence recognition, and this one involves lexical processing, and that's what we think those tasks do. So what we do is we propagate sort of up the ontology to the parents of those things. We use tools from the bioinformatics world. So people have done a lot of work in terms of comparing gene ontology annotations. We take those tools and use them to compare Cognitive Atlas annotations. So we can get what we call an ontological similarity measure. That's this Wang similarity thing. It's basically sort of how much overlap is there between the patterns in the ontology between these two tasks. And it turns out when we do that, it's certainly not as bad as I would have predicted. So the correlation between that image similarity measurement, the Pearson between the two images, and the ontological similarity measurement is 0.5. So we're capturing, a substantial amount of the variance in the activation from ontological similarity. So that's a starting point. It says that the ontology might actually be useful for something, but let's push it a little further and ask, can we actually, one of the things that we've always wanted to be able to do is say, given some new task that we've never seen before, predict the activation pattern that is associated with that task. And so that's what we're trying to do in this next analysis. So we build what's called a voxel-wise encoding model. And a voxel-wise encoding model is basically just a model that you fit separately at every voxel. This builds on ideas particularly coming from people like Jack Gallant and John Sorensis in the vision and visual attention literatures. The idea being that you try to fit a model that explains, what are all the things that voxel cares about. And so we do that separately. We take the data across those 93 tasks at every voxel, and we fit a model where the rows are those 93 tasks, and then the columns are all of the concepts in the Cognitive Atlas. So we basically say for each task, did it engage those different concepts in the Cognitive Atlas? In this case, we're not going to, here in this previous analysis, things sort of died off as they went up the tree. In the analysis I'm going to show you here, basically, if something is active, we just activate all of its parents 100%. So basically, you get everything above any particular thing in the tree is also going to be turned on. So that's why it's a binary matrix. So then we basically fit at every voxel a set of parameters. We get as many parameters as there are cognitive functions. So basically it's telling you, that's telling you for this particular voxel, how much does it respond to each of those cognitive functions. Now, there's more cognitive functions than there are data sets, so we had to use regularized regression. We used elastic net in this case. So how do we test this? We took a trick from, I think it was originally used by Tom Mitchell in his 2008-ish science paper on language processing. The idea, we call it a leave two out procedure. The idea is you have these 93 images, and you take all but 2 of them and you fit your model on them. And that's basically what you're going to use as your forward model. And then you take those other 2 tasks for which all you know is what cognitive processes should be turned on in those tasks. And you run the model forward, you multiply those cognitive processes by the weights at each voxel, and you basically get an activation map. You get a predicted activation map. And then what we do is we compare these predicted maps to the actual maps for those tasks. And our measure of accuracy is basically just, is predicted map for task 1 closer to the actual map for task 1, and is predicted map for task 2 closer to the actual map for task 2? If it is, we say we got it right. And it turns out that we can do quite well with that. We can get about 81% accuracy. Surprisingly, actually, I didn't think it would be anywhere near that high. This is well above what you get if you randomize the labels. It comes out to like basically 49.9%. And we're way above chance. And importantly, this relies heavily on the fact that we've sort of gone up the ontology and turned on the parents of these different concepts. If you only turn on the things that we annotated, so for example, if you only turn on visual sentence recognition and don't turn on any of its parents above it, you do much worse. You only get 56% accuracy. So this is sort of the first proof of concept that using this knowledge about the relationship between tasks is actually really useful for predicting something about brain activity. We can also look at which particular terms we do well or poorly on. So this is accuracy for all of the images that any term was associated with. And you see for some of them we do really well, like response execution, visual object recognition. Actually, surprisingly, working memory maintenance does well. Some that we do particularly badly on are things like cognitive control, updating. In part, this might have to do with how much data we have, right? But clearly, we have a good bit of data for cognitive control. We have 27 tasks that somehow involve cognitive control, yet we do pretty poorly. So there's a couple of conclusions you might draw from that. One is that we haven't annotated it correctly. Another is that maybe that's not really a useful concept. We'll come back to that in a moment. So just as an interim summary, we finally have some evidence that using an ontology actually gives you the basis for predicting something useful from cognitive processes to brain maps. Now, to move forward, one, we need a lot more data that's well annotated. And we probably also need a better ontology. And the question I want to turn to now is, how would we know if we need a better ontology? How would we know if ours is wrong? So this is a thought experiment that I've liked to use that I stole from Daniel Bubb. He wrote about this 20-something years ago. What if the phrenologist had fMRI? And so is everybody familiar with phrenology? So, phrenology was this sort of thing people did in the 19th century, sort of thinking that the bumps on your head had some relationship to the size of underlying brain faculties. And this was all based on, psychology that had come from that same period called faculty psychology, which basically said that the parts of the mind are things like inhabitiveness and vitativeness and destructiveness and alimentiveness and mirth and sublimity and so on. So, so what if they had fMRI? Do you think they would? So I hope we all agree that this is probably not the right way to chop up the mind, right? Had the phrenologists gotten their hands on fMRI, could it tell them that they were chopping up the mind incorrectly? What do you think would happen? So let's say that there's a lab that was interested in the study of benevolence. So what they might do is show people pictures of benevolent individuals and unbenevolent individuals and see what parts of the brain light up. And the anterior cingulate, among other places, would probably light up, and they'd get a Nature paper on the neural basis of benevolence, right? So, so that would be the first step. And then somebody, would probably say, hey, I have an idea. Young whippersnapper Franz Yarkoni would put together something called phrenosynth, right? Which would take all the phrenological fMRI literature and tell you, for example, this is the map for suavity, right? So what do you think would come out of that? Do you think it would look a lot like this? Exactly the situation that we're in now. So the question is, how do we know, imaging is almost certainly not going to tell us whether we're chopping things up right or wrong, or whether we've gotten it really wrong, at least in the way that we do imaging now. The question is, how could we do imaging in the future to do it differently? So I've already made that argument. So do we have any reason to think that our ontology might be wrong? And to answer that question, we have to get into the time machine. So we're going to go back to October 26, 1890, and ask what was going on in 1890s in science. And so in chemistry, there was still this big debate about whether atoms existed. Ernst Mach says, I do not believe that atoms exist. And that wasn't settled for 15 more years, right? In biology, people finally sort of agreed that cells exist. They finally agreed that disease was caused by germs and not by the miasma. But heredity still wasn't understood. And Mendel wouldn't be rediscovered for 10 more years. It turns out that there was computer science in 1890, but it was very different from today's computer science. This is the first tabulating machine built by Hollerith for the 1890 US Census. But we wouldn't have general purpose programming languages for more than 50 years after that. In neuroscience, people were starting to finally believe that the brain was made up of neurons rather than some big reticulum. What about psychology? So this is probably the most famous psychology book ever written, and probably the best. If you haven't read it, you can get it free online. You definitely should go read it. It's fun, and basically you'll come to a realization of what I'm about to tell you, which is that it's not clear how much we've really progressed conceptually beyond William James. So here's some topic headings from his book. The varieties of attention, the perception of time, forgetting, is perception unconscious interference, how the blind perceive space. Except for the way that they're written, these are all things that could be names of dissertations done in 2016, right? So that suggests that we haven't maybe had the same kind of conceptual revolution that other sciences have had. So how can we measure that? Well, one way we can measure that is to basically look at the terms that people use. So this is, I think, if you want to know what does the current conception of biology look like, the place to find that is the Gene Ontology. This basically is, a group of people has gone through and written down what do we think all the parts of a cell are, what do we think all the biological functions are, what do we think all the molecular functions are. And so the ontology sort of encodes all of that. So what I did was got my hands on some old biology textbooks. These are two textbooks from 1890s, one by Huxley and one by Parker. And I went through and looked at, they're both available through Google Books in full text. And so I went through and looked at what proportion of gene ontology terms actually show up in those books. And the answer is very few. And so, molecular biology didn't exist in the 1890s, so none of those terms show up. But cellular components, you'd think that they should have already known a lot of the cellular components, the parts of cells. About 1% of those terms show up in those two books, and they overlap pretty well. And biological processes, very few of them show up in those books. So what happens when you look at William James? So I took the Cognitive Atlas, which has admittedly a smaller number of terms than the Gene Ontology. Cognitive Atlas has about 800, almost 900 terms. Turns out that 22% of those, almost 23% of those, show up in William James's book. And when you look at them, they're all, again, things that sound very contemporary. Suggesting that maybe we haven't made, we certainly have not made as much conceptual progress as some other sciences like biology. Another way to look at this is just to ask what words were people using across the English language. So there's a tool called the Google Ngram corpus where basically they've gone through and for every possible ngram up to 5, so this is like sets of words up to phrases of 5 words, gone through the about 3.4 million books from 1800 to 2000 and asked how often does each of those ngrams show up every year. So basically you can ask what are people saying from 1800 to 2000 and how did that change over time. This is what Sherlock Holmes and Albert Einstein and Frankenstein. So I couldn't, I need to go back and do it for the full gene ontology. It's slow, and so I couldn't do it for all the gene ontology, so I did it on a random sample of small numbers, and you'll see what those look like. Here's what it looks like for the cognitive atlas. So this is basically from 1800 to 2000. For concepts that are either 1, 2, or 3 words, how often do they show up in the English language? You can see that for the one-word terms, they were basically almost all there by 1800. These are really folk psychology, right? These are heavily pre-scientific. Now, it's probably a little bit better, especially for the 3-word phrases, which really don't start coming on until the cognitive revolution, around the 1950s. And the 2-word phrases are, by 1950, more than half of them are in use. If you compare that to the gene ontology, you can see it's substantially different, right? There's much less sort of folk usage of terms from the gene ontology compared to the cognitive atlas. So that at least suggests that we might think about whether we can, how we might actually ask, where are we getting it right, where are we getting it wrong? Presumably, we have special access to at least some aspects of how our mind works, and so the fact that we're taking stuff from folk psychology isn't necessarily a bad thing. But in other sciences, they've almost invariably moved from sort of pre-scientific mode to through a revolution to kind of modern scientific mode. And it may well be that we need to do that as well. So how do we test that? And so I don't have results from an approach like this yet, but this is the kind of thing that we're thinking of. And we're starting a big project right now that in a couple of years, hopefully we'll have some data to address these kinds of questions. So let's say that, there are 2 different theories about the structure of executive function. And there's, everybody agrees that these 4 tasks measure executive function, and we can get activation maps for them. And let's say that I think that there are 2 parts of executive function. One's called inhibition and one's called updating, and they're measured differently by these 2 different tasks. But somebody else, importantly, this implies a particular covariant structure in the data between those tasks. It says that data from these 2 tasks should be more correlated with each other than with these, and these should be more correlated with each other than these. Okay, so it implies covariant structure in whatever. It could be behavioral data, imaging data, whatever. Somebody else thinks that, well, it's really just one big blob called executive function, and there's no difference between inhibition and updating. That implies a different covariant structure. And now we know how to test for covariant structures, right? We can use tools like meta-analytic structural equation modeling. We get observed covariance in brain data, behavior data, whatever data you want. And then we can actually compare those to the predicted ones and say which one's closer. Where do we actually get it right? And that can move us towards using lots of different types of data to actually inform our ontology. But it takes a very different approach than the approach that most people are currently taking in imaging. All right, so in the last part of the talk, I want to turn to some bigger questions about how confident we are in the way that we're doing science. And so I've already mentioned phrenology, right? And people laugh when they hear about phrenology, right? It's clearly they were goofy, right? I mean, they were goofy both in thinking that, like your jello of brain could push out on your skull, but more problematic, they were confirmatory in their approach, right? They would say, oh, you must be very suave. I feel like you have a big suavity bump. And the person says, yes, I'm very suave. And they say, oh, yes, so that confirms my theory of the suavity bump. And so we laugh at that, right? The question is what we do in our labs day to day qualitatively different from that in terms of its ability to find the right answer? And certainly there's growing concern about sort of across science, not just in neuroscience, but across science, about the way in which people do science today, particularly in sort of the bigger data world, might lead us to unreproducible or false answers. So I think the first shot in this battle came from John Ioannidis, who wrote this paper, Why Most Published Research Results Are False. An amazing paper. If you haven't read it, you should. But in the last few years, there's been, obviously in psychology, there's been a huge replication crisis. But we also see this in cancer biology and in other areas as well. So I think one can make the argument that neuroimaging is sort of a perfect storm for irreproducible research. And I'll come back and talk about a number of reasons why I think that's the case. I mean, you could like wring your hands and say, you know, I quit, or you can try to do something about it, and we're trying to do something about it. So we started a center a couple years ago called the Center for Reproducible Neuroscience that's trying to develop a platform and a set of tools to, one, sort of enhance people's ability to share data, but also provide them the tools to do more reproducible analyses. And so the way that I think about this is really in terms of kind of how do we design our scientific enterprise? Does anybody know what this is? It's a commercial paper cutter. Does anybody know why it has 2 green buttons right there? To prevent people from doing the really dumb thing of holding the paper up there and pressing the button and cutting their fingers off, right? So people who make paper cutters have built the paper cutter to basically anticipate the dumb things that humans do and prevent them from doing them. And in fact, that's what a lot of design is about, right? If you read a book like Don Norman's Design of Everyday Things, it's about, you know, how is good design related to basically preventing people from doing stupid things. And so I like to think about this in terms of a set of ideas that come from behavioral economics. This is a book from a few years ago called Nudge, which laid out this idea of what is now called choice architecture. The idea is that in any particular situation where we have choices, the default choices that one can make, or the sort of the things that are easier to do versus harder to do, are going to drive people towards making some choices more often than others. And so they've laid out this idea of a nudge, which is basically the idea of pushing people to do the right thing. When you think you know what the right thing is, pushing them to do it. And so some of the ways that they talk about using nudges are, one, just improving incentives, you know, sort of making sure that when people do the right thing, they get a better payout than when they don't. But also thinking about defaults. You know, defaults are incredibly powerful when it comes to decision making. If you look across European countries, there's a huge difference in organ donation rates between countries where it's opt-in versus opt-out. People, presumably there's no real difference in, you know, Belgium versus the Netherlands in terms of how people feel about organ donation. The question is, was the default to say yes or no? People heavily lean on those defaults. So to the degree that we can make the defaults doing the right thing, that's a good thing. And also providing feedback to people about what they're doing and sort of expecting people to do the wrong thing and sort of figuring out how to prevent those errors. Okay, so the first thing I want to talk about is power. So many of you are probably familiar with this paper from a few years ago by Button et al. That laid out, focused on structural imaging and also on sort of animal lesion studies, and made the claim that many neuroscience papers are woefully underpowered. The mode is less than 10%. We've recently done an analysis of the fMRI literature. This is sample size data from 2 different sources. The older one is from a manual meta-analysis that Sean David's group did and published a couple years ago. It's basically, they took a bunch of meta-analyses and pulled out the sample sizes from those. The blue dots and the blue line are automatically generated estimates of sample size from the Neurosynth database, where you basically go through and look for, in the abstract, things like 32 subjects or n equals 12 or things like that. What we see is that, you know, fortunately the sample sizes have been going up, and they're starting to actually get into a reasonable, you know, within the last couple of years, they started getting to almost a reasonable range. The median for 2015 was 43. Now the question is, what does that do for statistical power? And so, you know, your sample size obviously affects the kind of effect that you're powered to find. And one way to think about this is, you know, for any particular sample size, you can compute what is the effect size that you have 80% power to find. And there's multiple ways to do this. We've done it a couple different ways. This is for a corrected statistical sort of whole brain analysis looking for a single region. How much power do you have? And basically the median effect size across studies in 2015 that you have 80% power to find is about 0.6. That's a Cohen's d of 0.6. Now, if you're familiar with Cohen's d, the usual sort of you know, lore is like 0.5 is a medium effect, 0.8 is a quote unquote large effect. The question is, what are realistic effect sizes for fMRI? So we've also gone through using the Human Connectome Project data and asked for a number of the tasks there where you kind of have, you know, you have good expectations about what should be activated. what are the effect sizes in those regions? So we combined both anatomical regions of interest and functional regions of interest from neuroscience. So for example, we would take, for the motor task, we would take the motor cortex, and then we would go into neurosynth and do a search for motor and find the region that's activated in the motor cortex significantly across neurosynth studies for quote-unquote motor studies, and then conjoin those and just only look in the voxels that we see there. This provides us a way to independently come up with a region of interest and then ask what's the effect size within that. And the effect size is just basically the difference between the difference between conditions, which in this case would be activation versus rest for the motor task, over the standard deviation. And what we see is that for a couple of the tasks, it starts to get up into the reasonably almost large range. The supplementary motor cortex, this is the average percent signal change here is about 4%. So that's a whopping percent signal change effect, but still gives you kind of a medium-ish effect size in terms of the group statistics. And for tasks that have more reasonable percent signal changes, like the gambling task in the accumbens has a roughly 1% signal change, the effect sizes are down around 0.2, right? So what this says is that these are the kind of effect sizes that we need to be powered for, right? We're now currently powered to find effect sizes, the median's up around 0.6. If you're going to be studying anything, non-whopping, which probably means anything interesting, your effect sizes are almost necessarily going to be much smaller, and so that means that we're still underpowered to find most of those. Now you might say, well, power schmower, right? It's that I can run an underpowered study, and that just means I'm less likely to find something. And it turns out that that's the wrong way to think about it. This is a point that was first laid out in John Ioannidis's paper, and then this is from the Button paper where they have a nice sort of explication of it. If you run a low-powered study, you are less likely to find something, find an effect if it exists. But it's also the case that if you find something, it's more likely to be false. So think about it this way. Let's say you have 0 power, right? So you have absolutely no ability to find any effect. But you still have an alpha of 0.05, so you have a 5% chance of finding something. But everything you find is going to be false because you have absolutely no power to find anything. As your power goes up, the proportion of the findings that are true goes up. And so that's why power is not just about your ability to find something, it's about how believable the things you find are and how believable the effect sizes are. So this is showing that basically if you do an underpowered study and you find something, you're going to heavily overestimate the actual size of the effect. So one thing that I hear people say regularly is something to the effect that, well, my result isn't significant, so I need to add more subjects. If you ever hear anybody saying that, you have my permission to take a piece of paper and tap them on the head. And the reason is that this is a really bad thing to do. And this was shown by Simmons et al. In their false positive psychology paper published a few years ago, which basically showed that when you do this, because you're capitalizing on chance, right? You're capitalizing on the fact that, well, you might just get something popping over the threshold with a small sample, because things are really unstable with small samples. But if you had collected one more subject, it might go back down below the threshold. What they show is that when you do this type of adding subjects, when you don't have anything, it can lead to greatly inflated type 1 errors. And so the point that comes out of this is that one needs to always predetermine and write down your sample size before you start the study. And in principle, you'd like to predetermine it using some kind of power analysis. Now, you can't always do a power analysis, but you can take, for example, if you don't know what the effect size is, you can guesstimate, for example, from the stuff that I just showed you, that it's not going to be 0.8. It's probably going to be, for an interesting study, 0.3, maybe 0.2. It'll be small. And so that means that in general, you're going to need a lot more subjects than you thought you needed. Another threat to our ability to do good science is just the amount of flexibility we have in analyzing data. So we sat down with FSL and tried to figure out, what are all the different types of things one might do. And if you put together a factorial combination of these, you end up with 69,120 possible analysis workflows. That's more than the number of fMRI papers that have been published. And obviously, some of those people might think are kind of dumb workflows, but most of them aren't. There's no crazy options in here. And this isn't even changing the order, right? You can obviously double that just by changing the order of one of the two things. So an amazing amount of flexibility. Josh Karp did a study a few years ago where he took one of the data sets from OpenFMRI and ran it through 6,900 different pipelines. And what he found, this was one of our published papers, fortunately, the thing on the left, which is the mean, looks basically like the maps that we published in the paper, suggesting that, fortunately, we didn't get it too wrong. But what's really striking is the range across analyses of the Z statistic. So this is saying that in some of these areas, there's the difference between the highest and the lowest Z statistic across those 6,900 different analyses is 8, right? That's a monster difference in Z scores, suggesting that this really can make a difference. So, for a paper that, a review that we've been working on, we wanted to look at this in the context of multiple comparison correction, which clearly everybody agrees is a thing we have to do. So we took 100 papers, took basically the most recent 100 papers that come up in a search for fMRI and PubMed, and we found that 65 of those reported whole brain activation data. The good news is that only 3 of them reported completely uncorrected statistics, so people seem to have bought into the idea that you need to do correction. But there's a problem that I would refer to as a creeping problem. There's 11% of I think 7 out of those 65 papers basically did their analyses with SPM or FSL, but then used this other tool to do their statistical correction. This is Alpha Sim or 3D Cost Sim from AFNI, right? You might say, well, that's not a problem. Why is that a problem? It turns out that the p-values that come out of AFNI's 3D Cost Sim are badly inflated or badly deflated. They're overly liberal, right? And that's both because of a bug in the software. Many of you probably heard of, the kerfuffle about this Eklund cluster failure paper that came out a couple weeks ago, I guess. And they were the people who sort of pointed out, one, that there's a bug in the software, but probably more importantly, some of the assumptions in the software, both about the basically how it deals with edge effects and about the shape of the autocorrelation function, that those assumptions are incorrect, and so the p-values that you get out of 3D Cost Sim are way too low. And so we have to worry a lot when we see the field starting to use, and Jeanette Mumford and I have kind of privately kvetched in the last couple of years about how we saw this increasingly happen. Analyze your data using FSL or SPM and then use AFNI to do the cluster correction, and we wondered, there must be something funny going on here, and it turns out that there was. The field was moving towards a tool that gave them better answers. It turns out that those better answers were actually wrong answers. And so, fortunately, we have people like Eklund and Nichols to kind of figure out that it's actually, how it's getting it wrong, why it's getting it wrong. And so now we have, based on that paper, I think we have better ideas for how to move forward. So that's kind of a nefarious picture, right? This suggests people are out kind of trying to find trying to find something significant. And I think that's probably true even if people don't have it in mind, right? Our incentives in science are to publish papers with positive findings, right? Nobody wants to spend $30,000 on an fMRI study and have their grad student come in and say, oh, hey, I did our planned analysis and there's no significant activation, right? Nobody's happy with that, even though that's an outcome of science that we should be perfectly happy with. And so this is a paper by Andrew Gelman and Eric Loken published a few years ago that basically made the point that when you have choices in your data analysis pathway that are dependent on the data, you can get inflated p-values even without any sort of intentional p-hacking or trying to go fishing across lots of different things. That the idea is that there's this garden of forking paths. At every point you choose something that looks better than something else, and suddenly those add up to potentially sort of highly inflate your p-values if you haven't corrected for the fact that you've chosen lots of things based on the data. So it's taken me a while to come around to the idea of registration, of basically publicly register, or at least in a repository that can become public, like Open Science Framework, writing down before you do the study what exactly you're going to do, how many subjects you're going to run, what are the analyses that you're going to be, what you're going to do, what are the particular variables that you want to measure, and then doing those analyses when you're done. That doesn't mean it's the only things you can do, right? If we didn't do any exploration, we'd never find anything interesting, probably. But the point is that right now, when we write papers, we have to write the paper making it sound like all that exploration that we did was actually planned. This is this thing people refer to as harking, hypothesizing after the results are known. So the point is that we should be able to write a paper that says, hey, look, this is the things that we planned, and then we did or didn't find anything there. We went and did this other set of exploratory analyses. Those are, you should probably take with a bigger grain of salt, and we probably want to try to replicate before you really, go start treating people on their basis, or doing whatever else you want to do with them. But papers need to be much clearer about kind of what was planned and what was exploratory. So part of what we want to do in our center, and it sort of draws from this NITP workflow management project, is really thinking about kind of mapping that garden of working paths, right? So the idea is that you'd like to know, when you're done with a set of exploratory analyses, how many different things have you tried? What all have you done? It's easy to forget when you're kind of changing and tweaking things in an analysis. What all have you done? So, so one of the things that our platform will do is actually let you track all the analyses that you did. And another thing that we want to do is quantify this idea of kind of how much do things change when you change the analysis. So John Ioannidis has referred to this concept as vibration of effects. The idea is that, how much do the effects vibrate when you change the analysis? So one of the things that we're planning on doing is kind of allowing people to run their data through many different analyses, but instead of taking the best one of those many, you ask, how reliable, are my results across those many, and which, of the particular results that I have, which ones are more or less sort of reliable across analysis pathways? Where obviously, you'd only want to do this with analysis pathways that make sense and that are good. So the idea is you'd have a small set of curated workflows that you would run things across. So the point is that, we all need to focus on sort of coming up with science that we really believe is generalizable, rather than kind of treating, p less than 0.05 as hitting the jackpot. So another thing that's really important is sort of writing down what you did, right? We have this idea that in science we should be able to replicate a paper from reading it. We know that that's not true. And so part of the point beyond that is, actually sharing code and sharing everything that went into doing a study. But it's pretty astounding that, even after a lot of effort in the field, when we were looking at these papers, 22 of those 65 papers that did multiple comparison correction, we couldn't tell exactly what they did. They said something generic like cluster-based correction, but they didn't say what tool it was based on, what particular software they used, and so on. So recently, the Organization for Human Brain Mapping put out a report called the Kobodas report that's available on their website, and it'll be coming out in a short form at some point in the near future as a paper. And the idea is basically it lays out what are all the things that a paper needs to describe. And hopefully in the future, there are people now working on tools to actually automatically generate method sections, right? Our software knows about what we did much better than we do, right? We can't remember everything that was done, don't know all the particular settings in the software and the defaults. So we should be able to just say go and have a piece of software actually spit out a detailed or a non-detailed description of what we did. And I think that there's Yolanda Gill at USC is currently working on a set of ideas around data narratives that I think are going to sort of allow that. How am I on time? It's 2:25. Okay. So finally, I want to talk about sort of how it is that we tell people what we did. And this is a great quote from David Donahoe, who's one of my colleagues at Stanford. An article about a computational result is advertising, not scholarship. The actual scholarship is the full software environment, code, and data that produce the result. So Susan mentioned the MyConnectome project where I scanned myself many times, and we spent many months analyzing those data. And at some point, a lot of the processing of the rest of the data were being done by Steve Peterson's group at WashU, and at some point I got an email from Tim Lauman, who's the grad student I was working with, saying, oh, hey, we got an improvement to the surface-based analysis thing. I have a new version of the data for you. And I was like, you mean I have to go back and redo everything? And so that convinced me basically that to try to actually build a completely reproducible workflow. First on just my machine, but ultimately actually on a machine that anybody can run. So what we've done is so I don't know if you're familiar with the idea of a virtual machine. The idea is that using software like VirtualBox, I can run kind of a little version of a Linux box inside my Mac or inside any other computer. Vagrant makes it just really easy to set those things up. So what we did was we created a little virtual machine that when it's started up, it looks at this Vagrant file and it says, oh, I need to download all these data and all these software packages, and then I need to run these particular analyses. And so basically you go to, if you go here to my GitHub page, my connectome-vm, you can download this thing, and some number of hours later, depending on how fast or slow your laptop is, you'll have all the data from the study and all that went into the paper and all the figures that are in the paper, and you can see exactly how those figures were generated. And also have a browser for the data. And so I think that we need, the problem is that this was a ton of work. I mean, it took me a month or so just to do the work of, kind of getting all this stuff encapsulated, figuring out what the dependencies were. But it also saved me from another problem. So I'd first done everything on my Mac and had a set of results and wrote the paper. And then I wanted to take everything over to Linux in order to test it, just basically to make sure, because I was just going to share the GitHub repository. I wasn't going to do the virtual. I hadn't even thought about the virtual machine stuff. So I put it onto the supercomputer down in Texas and went to run it, and I got different results. From some of the statistical analyses. And so I was like, what's going on here? It turned out that one of the R packages I was using gave you different results on Linux versus Mac or Windows. And it was weird because it depended on whether you scaled the data and which was the X and which was the Y variable. It was a really funky bug. But it showed basically that just sharing your data is not enough, right? If I had just shared data and code, if I just shared the data and code, invariably somebody would have downloaded it and said, oh, hey, look, I'm getting a different result than you. What's going on? And if that had been different from the result that was in the paper, then that could have been a problem. And so this allows us to be able to give people exactly the way that the data were generated, so you could see the same numbers and the same figures. All right, so I think we've come a long way. We've certainly learned a lot. Hopefully we've learned, I don't know that we've learned $200 million worth or whatever it's cost to collect all those data, but we've learned a lot. But I think we need to move on to sort of new approaches now. It's clear to me, at least, that the way that we've been doing fMRI for the last 20 years has sort of hit a wall in terms of what it can tell us. And I think we also need to think about kind of how we can build our scientific enterprise to prevent us from fooling ourselves. How we can kind of redesign the choice architecture of fMRI to sort of kind of drive us to do the right thing and keep us from fooling ourselves. And finally, I just want to thank people who've given us money and people I've worked with on this, and thank you.